It's not like, yeah, get the HCl to stick to the beef. The whole thing. Usually I don't.
No, as long as your name's on that. It's fine.
Yeah, sorry.
Sorry. Like, go. More fun.
You like it?
You're kind of like
Good afternoon, everyone. Let's begin. I'd like to start with prayer, and consequently, I'm looking for a volunteer. Is there anyone willing to do that for the class? Please. Amen. Thank you. So quiz 1 is due today by 5 p.m. Let me review the options for turning this in. Some have brought their quizzes to class. That's great. You can leave it here. You can drop it off at my office, which is in the Benson building on the second floor, C201. You can simply slide it under the door if you wish. You can leave it with the secretary in the department offices. She will place it in my mailbox. You can also, there is a drop box for our class. It's in the main east-west hallway of the Benson building. It is just to the left of the large glass doors on the north and south of that main hallway. There's a display case, and next to the display case there is a wooden cabinet with slots, and we have three slots, 3 drop boxes for the 3 sections. You don't have to worry about which section you drop it in. they'll all go together. And you can also attach it as an email that can be sent to me. I ask, though, that you send it to my departmental email, swgraves@chem.byu.edu, and not to Learning Suite. I don't monitor Learning Suite. It may be that things will go missing, not missing, but will simply not get looked at for a period of time if they go there. Are there questions just about the mechanics of turning in the quiz? I will be having office hours today starting at about 3:10. I've had a number of people visit with me about aspects of the quiz. I won't give you the specific answer, but I'm happy to discuss your thoughts and maybe give you some guidance if you're having a problem. Yes?
How are we to get feedback after the quiz is graded? Like, are you going to pass it back to us, or how does that work?
Yeah, so most of these will be turned back, and underneath the display case in the Benson building, there are bins that are alphabetized, and they will be returned based on the last name of the first person that appears on the quiz. So if you have a series of 5 people, then it's the last name of the first person who appears on the quiz. So, other questions? Any questions from people online? All right, then today we are going to, we talked about 2 specific types of secondary structure, the alpha helix and the beta sheet in the last class. And today we're going to begin by talking about 2 fibrous proteins that have secondary structures that are dominant. That is, they effectively are also the 3-dimensional structures of these particular proteins. And I'm going to try to go back and forth between slides and some work in front of you here on the desk. So let me go here. Okay, so just putting things in context, we're going to talk about protein shapes, and the final shape of a protein is its functional state. If you lose that shape, if it unfolds, if it denatures, you lose function. We're going to talk about how secondary elements contribute to the overall tertiary structure. The tertiary structure is the final overall 3-dimensional shape of the protein. This is also called its conformation or its native conformation. And we're going to talk about these. We're going to talk about these 2 fibrous proteins, keratin and collagen. And then we're going to talk about ways of combining alpha helical structure with beta sheet structure, or multiple forms of alpha helical or multiple forms of beta sheet to form unique elements within a protein. And these are referred to as supersecondary structures. Okay, so let's begin with keratin. Keratin is a fibrous protein. By fibrous, it means that it occurs in long linear arrays. It is also a structural protein. Our hair, our fingernails are composed primarily of keratin. The keratin protein is almost a perfect alpha helix. That is to say, it forms this spiral shape The spiral is fairly linear. It conforms to all of the dimensions of the alpha helix, that is 5.4 angstroms per turn along the central axis, 3.6 amino acids per turn. They are spaced such that the R groups are positioned outward. They're about 100 plus or minus degrees away from one another, and they're also stacked so that they are above and below one another. There's hydrogen bonding between elements of the peptide backbone. So the peptide bonds will hydrogen bond one with another that is 4 amino acids further along. Okay. Now let me see if I can get back to where I want to. Okay. Okay. So let's say now I'm going to use this. It's one of the few demos that we have in our class. This is a single copy of keratin. Okay, you with me so far? And it forms this very smooth alpha helical structure. Remember that the alpha helix is a right-handed spiral. That is to say, if your thumb is placed along the central axis, the protein will rotate in a right-handed fashion around that central axis. But in the final form of keratin, it turns out that 2 copies of keratin, so this represents 2 separate copies of keratin, they are formed into a coil, what's referred to as a coiled coil. And in this case, these 2 strands are twisted about each other in a left-handed fashion. So single copy, a right-handed alpha helix, but you bring 2 copies together and they are twisted about each other in a left-handed fashion. So this represents now a coiled coil of keratin. Remember now that the amino acids are projected out from the central axis, and it turns out that many of the, or a fair number of the amino acids in keratin are cysteine molecules with that sulfhydryl, that SH group, projected outward. What you have happen then is that you can actually form disulfide bridges between 2 of these coiled coils. This gives additional strength to the keratin structure. So if someone has very straight hair, then they have fewer of these disulfide bridges. If one has very wavy hair, one has more of these disulfide bridges. If you're talking about a fingernail, there are even more of these disulfide bridges. And in the hoof of a horse or the horn of an animal, you have really substantial numbers of these disulfide bridges, giving additional strength to the overall structure. So these fibrous proteins have mechanical strength, especially in those tissues such as the fingernail and in horns and hooves of animals. Okay. So this slide then is meant to show that we have a single copy of, at the top, single copy of keratin protein. It is a right-handed alpha helix. Then we form this coiled coil with 2 copies of keratin. And then we begin to compile these. We bring them up parallel to one another, and we form these filaments and fibrils. And if you look at a hair, you will see these clusters of these coiled coils, fibrils, and filaments making up the interior of the hair. This is a little bit like a cable. Let's say you're building a suspension bridge. They use this same approach. They have coils, coiled coils that are turned in one direction, and then they will have these filaments where they will turn them in the opposite direction. This gives additional mechanical strength to the overall structure. You find this in rope as well. If you look at it carefully, you'll have things turned in one direction. You'll bring several strands together and they'll be twisted in the opposite direction. Okay, so this is keratin. Are there questions about keratin? Yes?
When you were talking about those disulfide bridges, are those between the two alpha helices when they're in the left-handed, or that's in the alpha helix itself?
No, it's not within the coiled coil. It's between coiled coils. So if you look at this slide, let me get something here. The disulfide bridges would be between copies of coiled coils. So you could have them going in multiple directions, but you're going to form these you're going to form these disulfide bridges between coiled coils. Now, if you're thinking about just the coiled coil, remember that the sulfhydryl groups are projected outward and not inward, so there is really no opportunity for the formation of disulfide bridges there. Yes?
When I was reading the textbook, they were talking about keratin in hair and how that is involved in curling hair.
Yeah, okay, so let's talk about getting a permanent. So you go to your local hairstylist and you request a permanent. So what they do, first thing they do is they treat your hair with something smelly. It is a reducing agent, specifically mercaptoethanol. And what this brings about is a disruption of the disulfide bridges. Then you take the hair and you loop it over a roller. You roll it up, and then what happens? So, do we have somebody who's had a permanent? Yes.
I haven't had one, but I'm guessing the disulfide bridges like reform with time.
They do, but how do they facilitate this? What do they do to somebody who has their hair in curlers? Yes. They don't actually, but other than air, but what they do is they blow warm air over the hair. And so this facilitates the reformation of these disulfide bridges, but we are forming them now in such a way that it reinforces the shape of the curler around which you've wrapped the hair. With time, these bridges will get broken, the hair will break or whatever, and you'll lose the permanent, but it will be there for many days. Other questions? Yes?
Would you recommend that we know the dimensions of keratin 3.6 amino acids?
You will need to know the dimensions of an alpha helix, right?
Is that standard for all alpha helices?
It is the way in which one recognizes an alpha helix. It has those dimensions, and keratin, except maybe for the ends of these keratin proteins, are in the form of an alpha helix. So this secondary structure actually becomes the tertiary structure. The final protein shape of a single copy is an alpha helix. So it is different than most proteins that we'll consider hereafter. Yes?
So when you get a split end, is that when the coil unfolds from itself?
Probably it's more the fibrils sort of uncoiling. So if we go to this, when you're talking about split ends, you're talking about larger collections of things. A single coiled coil is not going to unwrap, and coiled coils next to one another will break apart, but in usually large enough numbers, okay, that you can actually see the split end. A single coiled coil you would not be able to visualize, okay? Not with your eyes, okay? Other questions? Okay, let's now talk about collagen. Collagen is an extremely important protein. It is the most abundant protein that is found in nature. It makes up or it participates in the formation of a number of different structures within our bodies. It is the organic part of bone. So you have the collagen fibrils that form the bone, and then the bone gets mineralized. That is, you begin to lay down something called apatite. It's calcium apatite. It is the mineral that comprises then the bone. So, but you start off with this sort of scaffolding of protein, then you form the bone around these protein structures. It is also found in cartilage, it's found in tendons, it is found in the eye. So it is an important protein. It has its own unique secondary structure and its own unique sort of tertiary structure as well, quaternary structure. In the case now of collagen, it has a particular sort of arrangement of amino acids. So if you look down here, if you're talking, except for the end pieces, we have a glycine. It's probably not a good way to highlight that. We have a glycine every third amino acid. Okay. Now you remember that glycine disrupts, ends, terminates alpha helical structure. So we're not talking about alpha helices. Collagen has its own unique secondary structure called a collagen helix. And it differs quite dramatically from an alpha helix. In the first instance, it is a left-handed helix. Secondly, it has different dimensions. It is more elongated and thinner than an alpha helix, such as keratin. It is not a smooth spiral as found in keratin or other alpha helices, but it is a very kinked and crimped structure. And in the end, you take 3 copies of collagen, each with this left-handed collagen helical structure, and you're going to form a coiled coil by twisting them around each other in a right-handed fashion. It's also important to mention that frequently, about a third of the time, so we have a glycine every third amino acid for all practical purposes, at least in the general structure of collagen. But about another 25% of the amino acids present is a proline or a hydroxyproline. Now, proline is enzymatically altered to be 4-hydroxyproline. And so very frequently then, let me point out that you will have a proline or hydroxyproline as part of this triad, this repeated sort of structure. Now, the X can be any of several things. We'll talk about what those can be in a minute. Let me go here. So let me give you a sort of a demo. So if this is a single copy of collagen, it has a much more, it is a left-handed helix, but it is not a smooth spiral. It is really quite crimped. In forming, as we have collagen in its functional state in tissues, we take 3 proteins, 3 copies of collagen. Each of them has this unique left-handed structure, and we're going to form a coiled coil by turning them in a right-handed or twisting them in a right-handed fashion around each other. And so again, this gives us this very much stronger material to work with. And you'll realize that collagen plays some extremely important roles. Bone has to bear weight. Bone should not fracture easily. We have a lot of collagen in our skin. It helps prevent the skin from tearing. We have these fibers formed into cartilage. They need to be somewhat elastic, but they also need to be able to have a lot of wear and tear without rupturing or breaking apart. All right, now, yes?
Just to make sure I'm understanding this correctly, would you say that collagen and keratin have almost opposite structures?
I wouldn't say that. I think that would be confusing. I would say that they have very different structures, despite both being referred to as having a helical structure. The collagen helical structure is very different from the keratin, and it's not sort of the mirror image of it. So I wouldn't want to confuse somebody and think it's a left-handed alpha helix, and it's really not. It's skinnier, it's more elongated, it's more, it has these really acute angles within the collagen trimer, and so it differs in that way from keratin. Now, these 3 copies of collagen come together at glycines. Let me, oops. It's a little bit, it doesn't really show it very well here. But these 3 copies of collagen come together or pinch each at these glycines. So let me actually, I'm going to do that here. So you have something that looks like this. So the collagen actually looks something like this, and you're bringing 3 copies together at these glycines. So every 3rd amino acid is glycine, and that is where these 3 copies will touch together. The glycine has that very small hydrogen as a side group. It's able to adopt a very narrow angle, a very acute angle, but it also has a fair amount of flexibility. It's able to accommodate these 3 coming together to form the triplex collagen. Okay. Now, it turns out that there are yes?
What do you mean when you say that it's like glycines that they come together? Is there some
There is, sort of. It's hydrophobic in nature. But you have these three strands, okay? Let me go. You have these three strands, and where they touch together, that is a glycine. So where they all come together, that is a glycine. And then they are going to go their separate ways, but when they come back together, that will be at another glycine. Okay, so the point of contact between these three copies of collagen is always a glycine. Yes?
So this structure allows those pyrrolidone groups to be farther away from each other.
Yes, so yeah, so your proline with its ring structure is going to be, so let's just take one of these. So we have it in this, here would be, let's say, your proline or your hydroxyproline. It's going to form this very acute angle. Remember you have almost no flexibility in the phi and psi angles that are formed there. They are prescribed by the cyclized amino acid. And so you're going to, wherever that occurs, you're going to get a crimp in this strand. The glycine, on the other hand, is more flexible. It can open and close somewhat, at least those phi and psi angles can. And that's what allows three copies to actually fit together, okay, as you form the trimer, the trimeric form of collagen, which is the functional structural form of collagen. Okay. Now, it turns out that there are many different kinds of collagen. They are produced by different genes in different settings. So we talk about bone. Bone has a particular type of collagen, but as you can see here, there are multiple types of collagen that are designed and elaborated, produced in those tissues where they are needed, and they have slightly different functions. All right, so as we look at this, again, maybe you can see that you can see that there are really dramatic angles here. We're talking about quite acute angles in this. This differs quite a bit from the alpha helix, which is a smooth and gradual spiral. Maybe in this, if you look at the second structure here, the B structure, you'll get a sense that this is really quite bent in multiple places as it is elaborated. You have some?
Yeah, they just want to know if you need to memorize the 5 types of collagen.
They do, but nobody else does. Okay, no, no. It's I just want you to be aware that there are actually specific kinds of collagen found in specific tissues. They're designed, I mean, they're coded for by different genes, elaborated through different processes. It's really, you know, you could spend a semester on it by itself. So no, they don't need to know. Yeah. Yes?
What effect do the kinks have on the strength of it? Is the collagen stronger than keratin?
I don't think the abrupt angles, abrupt pinches and turns and twists of a single copy of collagen really dramatically affect its strength. I'm not sure that inherently it is stronger or weaker than keratin. However, having 3 copies together, I think, makes it slightly more durable. But there's an additional feature that we haven't talked about that we'll come to in just a minute. Now, I did want to point out again So here we have our three in C. You can see that we have now this this trimeric form. One of the things that I'd like you to look at and realize is that it's very compact. Okay, if we're talking about glycines, prolines, hydroxyprolines, and so forth, we're talking about many hydrophobic groups. And so the forces that hold the collagen helices together are primarily hydrophobic interactions. Okay, there is no hydrogen bonding within a single copy of collagen. So unlike alpha helical structure, keratin, where you have hydrogen bonding every 4 amino acids, there is no hydrogen bonding within a single strand of collagen. Nevertheless, these structures hold together very nicely and allow for collagen to be very hydrophobic and not dissolve in water. So that's good. Yes?
So you said there's no hydrogen bonding within collagen, within like one protein collagen, but also with the trimer, there's not hydrogen bonding as well?
Well, again, the amino acids are sort of oriented outward. It's not to say that there couldn't be some hydrogen bonding. It's conceivable, but it is not a major feature of collagen. Now, strands of collagen, these coiled coils, are actually coupled together through peptide bonds. And these bonds are formed from lysines. So lysines projected out from our coiled coil, and the lysine is acted upon enzymatically. It is oxidized first to a hydroxy species and then to an aldehyde. And so you have, we have here at the top, we have 2 copies we have our 2 coiled coils, 2 trimeric forms of collagen, and we're now going to oxidize these lysine groups. This produces now aldehydes, and these aldehydes will undergo a condensation. A aldol condensation is one of the 2 ways that these can react. And in doing so, you form this peptide bridge that holds these 2 coiled coils together. And this adds dramatically to the strength of collagen. There is another, this is again describing this reaction, this oxidation. So here's our lysine, undergoes oxidation to an aldehyde, and then there is this aldol condensation, which produces this type of bridge, peptide bridge, but this aldehyde can also react with an unoxidized lysine side group, so a primary amine, and it forms this aldamine structure, which is a second way of forming a peptide bridge between these strands of collagen. Okay, are there questions about this? You should pay attention to these reactions. You won't have to draw them out, but you should know that these peptide bridges form. They form from the presence of lysines that have been oxidized reacting with other lysines. Somebody have a question? Yes?
So these are both forms of those peptide bridges?
Yes.
Okay.
Yeah, both of these exist. Both of these happen spontaneously. That is, once you form the aldehyde, you can form these condensation products. Okay, there is a description here of how collagen is formed. I don't expect you to know this, but I provided to give you a sense of just how complicated this process is. Okay, if you start to think about it, these collagen strands can be enormously long. And you start to form these complexes of coiled coils forming fibrils and filaments, and these can extend the whole length of a long bone, the bone in your leg. And so how is it that cells can produce these very hydrophobic molecules and assemble them into trimers and then move them where they need to be? And it is not a trivial process. It is elegant and it is complicated, and it is not something I expect you to remember, but I would like you to have some respect for the bones in your body and the cartilage as to how it gets there, gets to where it needs to be in the first place. Okay. Let me comment on this slide. So in bone, you would anticipate that the fibers of collagen would probably be parallel one with another. And if you break a bone, bones break more easily in certain directions. And so it's easier to fracture a bone along the long axis of collagen. Now in cartilage, this could well be cartilage or the skin. Notice that our fibers are not placed in parallel, but they crisscross one another, and this gives increased flexibility, and it also means that skin, there's no susceptibility to tearing in one direction. Okay, so if you think of newspaper, if you try tearing it both ways, there is one dimension where the paper will tear easily, and it will tear in a fairly straight line. You don't want that to happen with skin. And so you form this meshwork, where the fibrils are crisscrossing one another, and you still have all of this peptide bridging between these different strands, and this gives flexibility, but it also reduces the likelihood of tears along one particular axis or dimension of the tissue. All right. Okay, we're going to quickly go through supersecondary structures. I don't expect you to memorize these or know these. I just want you to be aware of their existence. So here are some ways in which alpha helical structure or beta sheet structure are combined together. So multiple alphas, multiple betas, or combinations of the two. So here is a more complicated one. You can see that there's kind of a saddle or sandwich in this one. Now it turns out that when you have these supersecondary structures, it may not be surprising that you have some alpha helical and you have some beta sheet, and they get combined together, and they kind of maybe neighboring one another as they form. But what you see in nature is that where these supersecondary structures occur, they have specific function. So the protein has a function, but often these supersecondary motifs have a unique specialized function for the protein. So this one talks about immunoglobulins. Okay. Here is the helix turn helix. So we have two alpha helices. This is a common motif that is used for transcription factors. These are proteins that interact with the coding, with the upstream non-coding region of double-stranded DNA, but they regulate the transcription rates of the DNA. So they can either suppress gene expression or they can amplify gene expression depending on the protein. So this protein then has this element, it interacts with the DNA, and it causes a change in the expression of gene products. Look at this one. So here we have this, it's called an alpha-beta barrel. You can see elements, the interior staves of the barrel are beta sheet, but connecting two beta sheet pieces are these external alpha helices. This kind of barrel plays an important role in such things as movement of materials through a cell wall, or cell membrane, I should say. So ions, ion channels, things of this nature often rely on these kinds of barrels. This is, again, a protein that has multiple different motifs, different supersecondary structures. Each of these has its own unique role in the overall function of the protein. So you can see potentially, oops, this region right here is called sometimes a propeller. It kind of opens up like blades of a fan. It forms somewhat of a concave surface. And the dimensions of this and the side groups that are presented on this surface interact specifically with other molecules. Okay, here's another one of these propellers, a very elaborate, complicated one. Remember now that protein folds just sort of spontaneously, doesn't, it's just accidental, the final form that it takes. But these structures play very specific roles in protein function. Here's some more channels, some more of these barrels that are formed. These are mostly beta barrels. And again, represent channels through cell membranes, allowing for various entry or exit of materials into or out of the cell. This is green fluorescent protein, also a barrel. Okay, so there are many of these different structures. I don't expect you to know them, but I want you to understand that combinations of alpha and beta structure can form very elaborate, very complicated supersecondary structures, and almost always the supersecondary structures represent an active site on a particular protein. Sometimes there will be multiple active sites on a single protein, each of them carrying out some specific role or function. Yes?
So how do we differentiate supersecondary structures from like a tertiary structure?
Well, the tertiary structure is the overall structure of the protein. So let me, if you have something like green fluorescent protein, look up here, you have some just random coil, okay? It's not alpha helical, it's not beta sheet, it's not part of the barrel, but there it is. It represents part of that protein. And so the overall structure of green fluorescent protein would have this interesting barrel here, but then it has these other features that are on the barrel. If we go back to something like this, you'll notice that here's the most easily recognized supersecondary structure, but we got all of this business going on over here, including alpha helical structure, some random coil. So in the end, the overall three-dimensional conformation of the protein involves secondary structure, random coil, and these supersecondary structures as well. Okay, other questions? Yes, go ahead.
So there are just a couple questions about the quizzes. For the quizzes that are emailed in, when will they be returned, and how do you get feedback on them?
Well, they will be returned. As to when they'll be returned, it remains to be seen. It requires us to grade them. It's not as if, you know, it's multiple choice, and so we can run them through a machine. So it's going to take some time. But they will, in terms of feedback, I don't know that there'll be lots of commentary on each individual quiz, but I will provide a key. And so students should be able to compare their answer to the key and hopefully figure out what, how they are similar or different. Okay. Did you have a? After I've written exam 1. So, till I know what the exam is, it's hard for me to tell students what's on the exam. So. Okay, let me give you a quick sort of story. One day when I was at home, my wife said to me, you know, we spend a lot of time complaining about our blessings. And I said, what? I don't think I understand what complain about our blessings. She said, yeah, you know, for example, okay, what do you drive? Well, at that point, I was driving an '89 Toyota Corolla with 299,000 miles on it. The headliner had fallen down. I had it held up with magnets. Some of the windows could be rolled down. Some of them you couldn't. There were lots of extra holes in the body of the car, so I had lots of extra ventilation. But it ran, you know, and it got me to work, and it got pretty good gas mileage. So she said, okay, where did you serve your mission? I said, Thailand. I said, how many people had a car in Thailand? I said, wow, probably substantially less than 1% of the population had a car that could run. In Thailand, you know, you were doing pretty well if you had your own bicycle. It sometimes meant that people had to save up for a substantial period of time to manage that. But to have your own car, I mean, that was a real status symbol. And many of the cars that I saw in Thailand were very old cars that very often were well maintained, but still were very old cars. So I said, oh, okay. Yeah, if I am envious of my neighbor's brand new BMW, yeah, okay, maybe I look at my car and I'm not very appreciative. But if I think about the situation throughout the world, I should be grateful to have a car at all. And the same was true for food. I mean, many of the people I met in Thailand, they were fortunate to have one good meal a day and have a single set of clothes that they could wear. And I got stuff that's hanging in my closet that I haven't worn for a year. So anyway, maybe as we go through life, maybe we should spend less time complaining about our blessings and be more appreciative of those things that we do have. Thank you very much. I will have my office hours today as soon as I can get back to my office.